The overall goal of this procedure is to perform targeted cell ablation of kidney cells in the zebrafish embryonic kidney, the pronephros, for the purpose of studying renal epithelial regeneration following acute kidney injury. This is accomplished by first microinjecting zebrafish embryos with a dextran fluorescent conjugate. During an overnight incubation, the conjugates are selectively absorbed by nephron proximal tubule cells. The next day, the cells are examined under epifluorescence and selected cells are ablated using a pulsed laser system. Later, morphological and molecular changes are assessed using histology, whole mount in situ hybridization, and immunohistochemistry.
The main advantage of this technique over existing methods, like acute kidney injury using the nephrotoxic antibiotic gentamicin, is that such treatments trigger catastrophic damage and are lethal, precluding the analysis of renal regeneration events over time.
This method can help answer key questions in the kidney field, such as the identification of molecular signaling events that occur during epithelial regeneration. We first had the idea for this technique when we wanted to develop a detailed analysis of renal epithelial regeneration.
In petri dishes, collect fertilized zebrafish embryos in modified E3 solution without methylene blue. Raise the fertilized embryos at 28.5 degrees Celsius until they are between 48 and 55 hours post-fertilization, when the embryo's renal system has begun to function. Before beginning the injections, prepare an agarose injection tray using a mold with triangular depressions to hold the embryos. Next, prepare standard microinjection needles and an injection solution to label the proximal tubule. Begin the experiment by preparing the embryos. First, remove the chorion from any unhatched eggs using fine forceps. Then, rinse the chorion debris from the petri dish and refill the dish with 30 ml of modified E3 solution. Second, anesthetize the larvae with 5 ml of tricaine. Make sure the larvae are completely anesthetized by checking for a touch response. If they are not fully anesthetized, they will twitch during the injection. Third, using a plastic transfer pipette, put the larvae in the mold. Position the animals with their heads in the deepest part of the depression and their trunks along the side of the well. Now, proceed with intramuscular microinjections by loading a prepared needle with 2 to 3 microliters of dextran fluorescein. Position the needle and inject approximately 1 nanoliter of solution into a trunk somite of each animal. The dextran conjugates are readily endocytosed by the nephron proximal tubule, enabling preferential labeling of this epithelial cell population. Aim for the upper portion of the somite and avoid the yolk sac extension, or the nephron tubules will end up disrupted, such as in this improperly injected animal. Once the larvae are injected, transfer them to a clean petri dish and rinse them 3 times with fresh E3 media to remove all traces of tricaine. Now, cover the lid with foil and incubate the animals overnight at 28 degrees Celsius. Before beginning this procedure, prepare immobilization media and keep some aliquots of it for immediate use at room temperature and keep the remaining aliquots at 4 degrees Celsius for long-term storage. Begin by anesthetizing the zebrafish larvae with 5 ml of tricaine in 30 ml of E3. Just as before, make certain the fish do not respond to tactile sensation. Now, examine the injected animals under fluorescence to select larvae with easily visualized proximal tubules. Transfer up to 12 larvae to a new dish containing the same concentration of tricaine. At a compound fluorescent microscope outfitted with a FITC filter, bright field lighting, and a laser, perform the ablation. Begin by transferring an anesthetized larva to a dish containing immobilization media and wait 2 minutes. Then, transfer the larva to a glass depression slide containing a drop of immobilization media. Gently position the animal dorsal side up with a fine probe and focus on the dorsal side. Now, using a calibrated, pulsed, micropoint laser system, ablate one of the nephrons, leaving the other intact as a control. You will see dispersal of the fluorescence as the renal epithelial cells are ablated. Then, gently transfer the animal to a well of a 12-well dish and rinse it 3 times in E3 media to wash away the methyl cellulose. Continue ablating nephrons until the first 12-well dish is filled. Then, anesthetize another 12 animals and repeat the process. Dextran was injected to preferentially label the proximal tubule. 3 days post-fertilization, a nephron of each animal was laser ablated. 1 day after ablation, regeneration is not apparent. 4 days after ablation, however, regeneration has occurred. Following cell ablation, in situ hybridization was used to analyze changes in fixed samples at single time points. This embryo was not subjected to ablation. In contrast, this embryo had a large stretch of tubule epithelium ablated. And this embryo had a short interval of tubule epithelium ablated.
Following this procedure, other methods like whole mount in situ hybridization can be performed in order to answer additional questions like the identity of gene expression changes during the course of nephron regeneration.
Don't forget that working with chemicals can be extremely hazardous. Precautions such as wearing personal protective equipment like a lab coat and gloves are recommended during this procedure.